Well, thanks so much, Emily. That was a fantastic presentation. And the next one will be as well. I am delighted to introduce our next speaker, Dr. David Lewis, the Benjamin Castleman Professor of Pathology at the Harvard Medical School and Pathologist in Chief at Massachusetts General Hospital. Dr. Lewis is a national leader in molecular diagnostics, and his laboratory was the first to demonstrate molecular approaches to subdivide gliomas biologically and predict response to therapies. In his session entitled Understanding Your Tumor, What, How, Why, and Who, the WHO, he'll provide a very simplistic understanding of brain tumor classification and why it is important. Thanks so much, Dr. Lewis.
So thank you for the kind introduction. I also wanted to personally thank the last two speakers who gave us a really superb talk. It's a tough act to follow, but I will try. As you heard, what I'm going to talk to you about is what goes on to analyze a tumor, why is that done, and how does that change over time? And the reason this is important is, as all of you know, the type of tumor determines the therapy and it determines the prognosis. And so figuring out in detail what type of tumor is there and what its characteristics are governs a lot of what happens with a patient in the subsequent course. And we'll talk about that as we go along. So I have no conflicts of interest at all for this presentation. What we're going to talk about are what are the changes that occur to lead to something like a tumor developing. By understanding what those changes are, you can then understand what pathologists do to classify tumors. And we'll talk about why pathologists classify tumors, and then the specific guidelines that they use, which is the WHO part of it, the World Health Organization part of it. And just this past year, there was an update to that classification. So if you think about what we do in our everyday life, and probably what all of us are doing now, is we're doing something. For example, we might be moving a finger. And to move a finger, it requires many different organs and tissues to work properly. To move a finger, you use your brain, you use the nerves, you use the muscles, you use the bones. But when those functions get diseased, you have trouble with that type of activity, moving the finger. So the question becomes, what is abnormal in a tissue, in an organ like the brain or the nerves or the muscles or the bones, to lead to a disease state? And the basic answer is that tissues, organs like the brain, function properly when certain molecules, certain things called proteins work properly. And I'll tell you a little about what these are, but basically they are compounds within a cell that do what the cell is supposed to do. For example, with the brain, it may be a protein that allows the brain to transmit an electrical impulse telling another part of the brain to do something. Proteins form when they get codes from another molecule called RNA. And RNA in turn is instructed by the master molecule, which is called DNA. And this comes together in structures called chromosomes and genes, which I'll show you. So if you think about it, when somebody comes into the doctor with symptoms, for example, to say my finger's not working well, the doctor will typically do a physical examination and examine that, and that gives an idea of what functions are working. But then when you want to look at tissues themselves, you have to use other technologies. And in the last lecture, you heard about some use of radiology to look to see whether something's normal or abnormal. And then also, Dr. Hervey Jumper showed you some examples of images obtained using a microscope of examining pieces of the brain. We have ways of looking at proteins. I won't talk in great deal about any of these. We have ways of looking at RNA. And then most recently, we have some very sophisticated ways of looking at DNA and chromosomes and genes. And the reason I emphasize these last ones, and we'll talk about these, is that these have had the greatest impact on classifying tumors, particularly brain tumors, over the last 10 to 20 years. And they've played a major role in the updates of the World Health Organization. So radiology, you understand, you've seen pictures where you can do an image and find a tumor in any organ in someone's body. Using a microscope is a way of taking a piece of tissue, for example, a biopsy given to us by a neurosurgeon, given to the pathologist, and we can look under the microscope and see something like this. The purple dots here are the centers of cells, things that we call the nucleus. And the pink stuff in the background is what we call the cytoplasm. It's the body of the cell, and that's what does the activities. So the purple, the nucleus, is where the DNA is, and the pink stuff is where all the proteins are carrying out their various tasks. We can look directly at the proteins. This is one example from a brain tumor where the brown there reflects a very specific protein found in those cells. And as I say, we can look at RNA and we can look at DNA using techniques called sequencing. And the one abbreviation you may hear when your doctor talks to you about it is NGS, which is next generation sequencing, a very powerful technique to do analysis of the DNA. And then a technique that has also become very powerful in brain tumors called Methalone Profiling. So what I'm going to do for you now is explain those two technologies. Recall that I told you that when you look at these cells under a microscope, for example, this brain tumor, the purple is the command center of the cell, and the pink is the action area, the body of the cell. Well, if you look at it in greater detail, this is a nucleus, and folded up in the nucleus in very specific ways are long strands of DNA. There are about 3 billion of these in every cell in our body. And each strand of DNA has a specific sequence to it. So this one says CTCGA, and another one would have a different sequence of those specific areas of DNA. And so that's why we call it DNA sequencing, because we can figure out what the sequence is of a particular strand of DNA. Now, in order to get all of this DNA into a nucleus, they're packaged in interesting things that involve adding methyl groups to the DNA. And I won't go into detail what a methyl group is, but basically it allows the DNA to be compartmentalized. And there are patterns that we can distinguish of those methyl groups. So we now use both of these technologies, NGS or DNA sequencing and methylome profiling, to classify brain tumors. And this is just a very simple, actually, example. Each one of these rows here, each one of these little strings is a different piece of DNA, and we've sequenced all of them. And then you see the actual sequences down here. And then we align all of these together. And what we see, lo and behold, in this example, is a piece of one chromosome labeled in red and a piece of another chromosome labeled in gray. This is not normal. It should be the same chromosome. And this tells us that in this example, which is a lung cancer, there's a specific change in the DNA. And this is an example of what we call methalone profiling, which is a very complicated technique. It actually uses artificial intelligence to do the clustering of tumors. This is many, many tumors. Each of the little dots here is a tumor. And you see how if you put DNA from one type of a brain tumor into the classifier versus another type, it will separate it based on those methalome patterns. So who does all these analyses? Well, the answer to that is straightforward. They are pathologists. Here you see 3 friendly pathologists working on one of these complex machines. But the picture that I like to use of what pathologists do is this famous one from The Wizard of Oz. You remember when Dorothy and her colleagues are finally getting to see the great wizard. He's this holographic image presented on a screen. It's extremely impressive. This is the great Wizard of Oz. But you remember the little dog Toto pulls aside a curtain, and there's a gentleman behind the curtain controlling all of the knobs and allowing this great Oz to appear. I jokingly say that this is the pathologist, the man and the woman behind the curtain that gives the answers to the great doctors, the oncologists, and the neurosurgeons who can talk directly to the patients about what their conditions are and what will be done to treat them. But it all relates to these analyses that go on behind the scenes by pathologists. The important point is that in the movie, they say, pay no attention to the man behind the curtain. And I wanted to say that, well, you can pay attention, because if you have questions about your tumor type, you can talk to your oncologist and your surgeon. But with those people's permission, you can also talk to your pathologist who can explain things directly. So why do we then go ahead and classify human tumors? Well, what we all know about is that it's important for the care of individual patients. One type of tumor has a different prognosis from another, and the therapies are guided by the types of tumors. When you start thinking about other uses, when you do clinical trials, the results of the clinical trials may be completely wrong if the tumors aren't classified properly. When you think about experimental studies in petri dishes or in mouse models, again, the results of those can be completely wrong if the tumors aren't classified. When you start thinking of large-scale studies to find out what causes certain types of tumors, that also requires accurate classification. And when you ask how governments and health insurers allocate money to support different types of healthcare, that also depends on the accurate classification of diseases. So as a result, when you change tumor classifications, you have to do it carefully and only periodically, because it has many effects, not only on the care of individual patients, but on population health as well. It's also really important to realize that all of these classifications are imperfect representations. They represent the state of the art, the state of understanding at a particular time. And they also represent the interpretation of limited numbers of people. They should really be thought of as works in progress. And so each classification improves on the last one and sets up for further improvements to happen in the future. For the World Health Organization, classifications have come out since 1979. There were 5 of them until last year. And this is the newest one from 2021, with a list of the various editors from around the world. But it's not only these editors, there were, for generating this, input from 199 experts from around the world. But to generate that book, it's not only dependent on the group of 199 experts, we actually have additional organizations that help us do this. So one of them that we started about 5 years ago is called See Impact Now. And we named it this because the WHO, the World Health Organization, only publishes their classifications every 5 to 10 years. And the pace of change in the field, because of things like DNA sequencing and methalone profiling, is so rapid, we didn't want to wait 5 to 10 years for new WHOs. We wanted to see impact right now. And so we have this additional organization working on this. And there's another organization I'll tell you about in a moment called the International Collaboration on Cancer Reporting. And these organizations work together at an international level to do these classifications. And this is just a list to show you the complexity of the See Impact Now organization, how many folks we have working from around the world on this. To get from the 2016 to the 2021, in fact, almost all of it was based on the advice of the See Impact Now group. The International Collaboration on Cancer Reporting, the ICCR, is also really interesting to know about, because you might ask, well, how do I know that my pathologist in San Francisco is issuing the same type of report as another person's pathologist in Boston, or another person's pathologist in Hong Kong or Paris, for that matter? So what this organization does is it issues guidelines to say, if you're a pathologist reporting on a tumor of the nervous system, you need to follow this exact format so that the significance of what someone says in Hong Kong is applicable to what somebody's doing in another city. So to summarize what I've told you, classification of tumors is done by pathologists. We use both traditional techniques like microscopes as well as cutting-edge techniques. And don't be hesitant. You can talk to your oncologist or surgeon about talking to a pathologist if you have more questions. The accurate classification of tumors not only affects the care of individual patients, but it has many other effects on research and on healthcare spending. And as a result, when we revise tumor classifications, they have effects on individual and population health. But importantly, these are imperfect. These really should be thought of as works in progress. The new 2021 classification does have many changes. We've added what we call new entities. They're really not new, they're just newly recognized, as well as new subtypes of entities. And this is in almost all of the sections for the glial tumors, as well as some of the more common ones found in pediatric populations. In fact, there were 22 new entities added into the 2021 classification of brain tumors. And unfortunately, given the scope of changes, there's really not time to talk about the individual ones here. Lastly, I really want to thank the ABTA. This is, believe it or not, me, a much thinner me. Almost 25 years ago, with my friend and colleague Greg Cairncross, when we won the ABTA Research Excellence Award. This is Naomi Berkowitz, who was the executive director of ABTA. So I've been thanking the ABTA and will continue to do so for a long, long time. And with that, I will end, and thank you for your attention. And if there is time, I'm glad to take any questions.
Thank you so much, Dr. Lewis. Just as a reminder, if you do have questions that you'd like to ask, please click the Q&A button on the right-hand side of your screen. Click the blue Ask a Question button, and then type and submit your question, and we'll answer as many questions as time allows. All right, so one of the first questions we have here is asking, how often is pathology testing done? Is this something that is only done at the time of diagnosis, or is it something that you repeat and is ongoing?
Pathologists will look at every single tissue taken out of a person's body. So every time a biopsy is done, every time a resection is done, or for that matter, every time you have a blood test or a urine test, all of that gets examined by pathologists.
Thank you so much for that answer. We have a question here asking, how can patients who might not have access to larger treatment institutions, how can they get access to this type of pathology testing and molecular testing that you discussed today? Are there any resources that you can share or places that you can advise patients that don't have access to those type of treatment centers and where they can turn to?
So I'll answer that in two ways. One is talking about the United States, and then talk about other parts of the world where there are fewer resources. In the United States, essentially all patients should have access to this through their hospitals, even if their hospitals are community hospitals. So when they refer a patient to a larger center, the tissues can come along, can be transferred to the larger center, and these molecular analyses can be done after the fact. So months or even years after your surgery, the tissue is still available in nearly all cases for additional analysis. There are also private companies that will do this kind of analysis very well. And so it should be available to anyone who does reach out to get it. And oncologists at smaller hospitals where the pathology department at that hospital don't have these facilities should know about where tissue can be sent for these analyses. In countries that really don't have this available, we're working internationally with a variety of organizations to come up with what we call surrogate markers, where you can use simpler technologies to get a fair amount of the way towards the same type of information. It may not get you 100% of the way, but it can get you awfully far.
Thank you so much. And I really appreciate you covering that international aspect for any of our attendees who are joining outside of the US as well. So another question we have here is asking, how frequently should pathology be updated as you're assessing any changes in tumor grade?
It's a question without a clear answer. Some people we think of it as a balance, and some people would try to tip the scale towards more frequent updates, and other people would tip the scale towards less frequent updates. So what do I mean by that? If you're a patient or a treating doctor, you want the latest information, and you're concerned about an individual patient, and what you want is the most up-to-date information. And so for those types of situations, you might argue that changing it every few years would be okay. On the other hand, if you're an epidemiologist looking for clues about what cause diseases, you certainly don't want classifications changing that frequently, because you want to be able to look over time, and you want to be able to know what's called an A tumor in 2022 has some relation to what's called an A tumor back in 2000, or what's called a B tumor back in 2000. So you need to be able to translate over time for some of these. And so those people would favor tipping the scales towards more stability in a classification. And every decision that we make with classification is taking into account that delicate balance. And it's also a delicate balance for making sure that if we recommend something at the level of the World Health Organization, the great majority of people around the world can benefit from that. It's really silly to suggest something that's only available in one medical center, around the world. That just wouldn't fly.
Thank you so much for that answer. All right, so another question we have here is, based on WHO classifications, Can you explain what the difference is between a grade 4 astrocytoma and glioblastoma?
Yeah, this is actually a very complicated question, and it's one of the major differences between the 2016 and the 2021. I'll try to explain it briefly, but it's complicated. In the past, a grade 4 astrocytoma was equivalent to a glioblastoma. So for 2016, that was an equivalent diagnosis. At the present time, glioblastoma, that diagnosis is only made if the tumor has a wild type, a normal IDH set of genes. So you can't make a diagnosis of glioblastoma in the 2021 unless you've done some DNA sequencing and established that the IDH genes are normal. So what is an astrocytoma grade 4 if it's not a glioblastoma? It's typically one where the DNA sequencing has shown an abnormality, what we call a mutation of the IDH genes. And that's then what we call an astrocytoma IDH mutant W.H.O grade 4. So both of those tumors can look similar under the microscope. They both get a W.H.O grade of 4, but they're biologically very, very different tumors. And we know that IDH status can affect how you go about being followed, how you go about being treated, prognosis, some therapies. So they're really very, very different. Historically, we lumped them together because we didn't know any better. And as we've discovered more, now we're trying desperately to separate those out because we just believe they're really, really different tumors.
Thank you so much for answering that question that I know is difficult to answer in such a short amount of time, but you did a fantastic job of helping us to better understand that. Another question that we have here is asking, from the time that the tissue is obtained, how long does it take for tumor types to be identified and classified?
It really depends on the technique being used and how readily available those techniques are in an institution. So, for example, in a large place like ours, we will know the IDH status of a tumor within a couple of days, often before the patient is coming back for a post-op visit. But if you have more complicated DNA-based analyses to do, or you don't have the technique handy right where you are, I'd say a realistic time frame is 3 to 4 weeks.
Thank you so much for sharing that. I'm going to take just one last question. Can you share a resource that may provide patient-friendly summaries of what the new WHO classification updates are? Because it can be a little hard to follow.
Yeah, it's a very good question. We've tried to do a number of summary articles, and these are mostly summary articles for doctors. Eventually, oftentimes there are summary articles put together for laypersons. Offhand, I don't know of any that have come out yet, but they should at some point be coming out. The other way to think about it as a layperson is to go to one of the summary articles that are written for physicians, and I can provide probably the most comprehensive one of those to date if anyone's interested. You can go to that and look at what it says about your particular or your family member's particular tumor type. And even though it's a large article and it's written for physicians, there may be enough in there about your tumor type that you'll get an insight into that specific part of it.
Thank you so much, Dr. Lewis. I think that is great advice. Unfortunately, that is all the time that we have for questions for this session. Thank you again for your presentation, and I'm going to turn it back to Emily.